This conference will now be recorded.
Okay, hello everybody, and thank you, Marine, for this kind introduction. So my aim today is to try to show you how chemometric tools may help you to integrate data from different sources with the aim to obtain a better characterization of your samples and, of course, hopefully get a better understanding of the biological phenomena you are interested in. I'm sure you all know that chemometric tools are important, and they are actually part of the knowledge discovery workflow in metabolomics and in other omics. Of course, the starting point is the production of high quality data based on the adapted sample preparation and data acquisition. Then you need to process the data with specific strategies that depend on your analytical device and the nature of the signals you are measuring. And very often this step is important because it helps to further improve the quality of the data that will be analyzed in the next step. So the next step is actually the data analysis part. And among the many solutions available to analyze your data, from classical hypothesis tests to more advanced machine learning approaches we hear a lot about these days, I think chemometrics offers efficient and complementary tools to explore your data and compare the experimental conditions you included in your design. And so I will mostly talk about this part today, but of course, don't forget that this is not the end of the story, because you need to put the results you obtain into a biological context to really gain knowledge about your system, and this can be done using bioinformatic tools, but this will not be the topic of my talk today. Actually, as you can see, all of these steps are important. But I think chemometrics is really at the heart of the knowledge discovery process, and it's an important part of the knowledge discovery process. So why do we need specific tools to analyze our data? Actually, this is related to the structure of the data we are now able to acquire with our analytical devices, because this ability to generate a large amount of data changed a bit the way we can ask biological questions. Because on the left side of this slide, you can see let's call it the traditional classical approach that starts with information collected from the literature and the prior knowledge in your lab, and based on an hypothesis-driven approach, you will try to bring experimental proof to in some way a quite targeted question, and which can be seen as a kind of reduction strategy, because you really target one question and you give one answer. This ability now to collect data in increasing amounts and complexity has changed a bit the way we can deal with this knowledge discovery, because now we can start by collecting data and using a more global strategy, try to build hypotheses from the data we collected. And this changed also a bit the tools we use to extract what is actually considered as the information within the data we collected. For that, I'm sure you perfectly know that there are multivariate analysis approaches that will help to reduce the dimensionality of your data, to help you to get a synthetic view of your data sets. Of course, you need to define the aim of the modeling strategy. It can be unsupervised with the classically principal component analysis, or a supervised approach when you want to go more in the discriminant analysis of your subgroups. But whatever the aim of your model, the idea is to get a summary of the variations in your data sets and get a model that is able to show some interesting variation in the data. And all those models, well, at least the one we are used to apply in metabolomics, are usually linear, because I think one of the main objectives of the modeling these data using our multivariate models is to be able to go back to the contributions of our variables. Meaning that, of course, you'll look at the score plots to be sure that your experimental groups are well separated, or that you can distinguish some meaningful pattern among your observations. But I really think that the added value of these multivariate models is the way we can evaluate the contribution of the variables, to rank them or to filter them, to in the end get some molecular signatures that may be useful to describe the phenomenon you're trying to investigate. And of course, depending on the aim of the model, you can end up, as you can see on the right side of this slide, you can end up with different point of view on your data that will extract different kind of information from the variables. You probably have used these multivariate models for a single data matrix, but today we can see that an increasing number of studies aim at the combination of multiple sources of data. And I think that this is mainly due to the fact that whatever the measurement technique, no matter how powerful or how good its resolution or sensitivity could be, most of the time it's not sufficient to get a complete picture of your samples, just because they are too complex in terms of physical-chemical properties, for example. So when you're measuring things with your analytical device, you are actually you get access to a partial and hopefully not too noisy, but not the complete picture of your phenomenon. And that's why people started to think that it may be very informative and very efficient to try to combine multiple sources of data with the aim, of course, to get a more complete overview of the samples and to get more insight into biochemical processes, for example. One of the main application field today, of course, obviously we hear a lot talk about systems biology that tries to gather different biological layers together. But this is not the only case. We could be interested in the integration of multiple data blocks. For example, if you want to collect metabolomic data from different cell types, different tissues, different organs, and integrate them to get a more complete understanding of the way the organism deal with different stimuli, for example. This may lead to this kind of data structure that needs integration tools to make sense of the different data that have been collected. We can also think about different stages to describe a process that can be either related to evolution over time or according to different exposures, doses. And maybe for people that are more involved in the analytical part of the metabolomic workflow, we can also think about this complementarity in terms of analytical techniques, because it can be very interesting to try to combine data measured with separation methods coupled to mass spec, for example, NMR. Of course, this is the topic of today's webinar, but also other spectral or separation methods. So this is also something that can be done with the tools I would like to show. To go more into an integrative understanding of the systems that you're trying to describe. What does it change in terms of data structure? Again, I think you're perfectly familiar with the two data structures that are associated with a vector when you collect a single response for a series of observations. This obviously leads to a data matrix when you collect multiple signals to describe each of the observations. Of course, classically, the data matrix is represented as a collection of values sorted according to the rows that are related to the observations and the columns that are the variables of your matrix. And when you collect data from different data sources, you will end up with a collection of data matrices that can have different number of observations. These observations can be the same or not. We'll see that later on. And these observations can be described by variables that can be the same or different variables. And again, the size of the data matrix can vary according to the measurement technique you used. So the first aspect to consider when you want to integrate your data is what possibility can you use to link these matrices to build relationships between either observations or variables. And here you can see that there are different situations, starting from on the left side. You can see that we can imagine to collect different data matrices that are obtained maybe with the same analytical device. So here you have always the same variables, but the observation are structured according to groups, and these observations are not the same. So the only way to connect the data matrices is through the mode of the variables, and this is called actually a vertical multibloc data structure. And there are algorithms, methods that are designed to account for the fact that actually the same variables were collected for different subsets of samples, observations, and you can build a model that will take these links into account. Maybe on the top right part of this slide, this is the situation you would think first when talking about multi-block data structure. In that case, you can see that we have different blocks of variables that are linked through the observation mode. And in that case, the nature of the variables can be very different. And again, there are models that are able to deal with this situation to account for the fact that the same observations have been measured through different data matrices. And again, multivariate model may help to get an overall picture of all the data you collected. When you can link the data matrices both with the observation mode here, the same observation are in the different data matrices, and the same variables, this is something we can very often get when you follow over time a phenomenon. Of course, the measurements need to be non-destructive because you need to follow exactly the same individual over time. And this leads to, again, another type of data structure, which is called a multi-way data structure. And again, there are specific algorithms that are able to deal with this kind of links. And at the right of the bottom of the slide, you can see that you can even have to deal with more exotic data structure with sometimes links that are related only to the variables. This is the case for the two first matrices. And here with this L-shaped data structure, and the second and the third matrices are linked through their variable mode. And there are also methods to that are able to handle this kind of relationships. So all that to say that the first thing to consider is to look at the structure of the data to assess which one of the modes we can use to link our data matrices. But whatever the structure, the aim of the data integration strategy in omics are of course to, as you would do with the single block analysis, to end up with molecular signatures. We hope these signatures to be more complete, of course, and to give maybe deeper insights into biological processes. Then, of course, you may be interested in the integration of all your results into metabolic networks and things like that to get more, again, more mechanistic insights into your systems. And I also would like to mention that I will talk today about extensions of multivariate models that are not that different from PCA and things and models like that. But I will also mention that there are other ways to integrate data sets. Today, we see an increasing number of approaches that are based on networks that try to gather information from different layers using networks. Bayesian approaches are also increasingly used to do that. And of course, with all these developments in machine learning, today a lot of efforts and research is done also to get some modeling approaches, strategies that are able to deal with these data structure. So all that to say that we will talk about matrix factorization. I think it's a very interesting tool because we will continue to build linear models that really provide a direct information and feedback about the contribution of the variables. If we think about data integration, actually we can distinguish, this is a bit theoretical, but you will see that it may be helpful to maybe see which tools you can use with your data structures. We can distinguish different levels of data integration. The first level we can see is called the high-level data integration, and maybe you already did this kind of data integration methodology. It consists in the analysis, the separate analysis of the different data matrices, and you may be interested to use multivariate models. I don't know, PCA, PLS. And you end up with a score, a subset of biomarkers for each of the models you get for each of the matrices you've been collecting. And then actually the data integration process is done by the experimenter that who will try to gather the information to make to make a biological interpretation of all the results you get with the individual models. So it's interesting because when the observation mode is not shared, you can still work with the different matrices separately and try to integrate the data, the results, sorry, at a higher level. The mid-level and the low-level involve a different structure. In that case, you need to get a common observation mode, because in that case, you will consider the data matrices together. The mid-level strategy consists in the extraction of patterns or subsets of variables from each of the data matrices, and then you concatenate them, and you can then apply a traditional PCA or PLS model to get again an interpretation of the global information in your data collection. But today I will mainly focus on the last possibility, which is the low-level data integration. I think it's really interesting for us because it's a data fusion strategy that really tries to connect the signals from the different matrices directly, rather than trying to extract something or compute components or before trying to connect the signals. And to make things even more complex, you will get different terms to talk about this data integration methodology with data fusion, multi-set, multi-view, multi-block analysis, and it's very often it depends on the application field. So this was a bit theoretical, but I would like to show you an example that we've done in Geneva a few years ago, which was a collaboration with one of the major flavor and fragrance company here in Geneva. And among all the products they make, they were very interested in their ability to characterize the lemon essential oils they use, because it's part of the wide range of their products, including perfumes, cosmetic foods, pharmaceuticals, and they were interested in the assessment of the quality and the chemical characterization of the products they use. So for that, they wanted to see how useful. Could metabolomics profiles could be used to get maybe more insight into the chemical complexity of these their samples, and maybe to to detect contamination, adulteration, or things like that related to the quality of their products. So for that, they started a sample collection campaign, and we were really lucky to work with these samples that were collected in different countries in the world. You can see that they collected samples from Spain, Argentina, and Italy, and they also included different producers in each of these countries. Obviously, here their question was related to the influence of the geographical origin and of the cultivars that were actually used in these companies, to see and to assess whether these differences may lead to composition differences in the end when it comes to the lemon essential oil. So this was the first question. What was the effect of the geographical origin? And they also wanted to get more information about the influence of the different extraction processes that are involved in this cold pressing of lemon oil. And they distinguish different processes with the FMC, sfumatrice, BOE, and pelatrice. And these processes were characterized by different way of extracting the oil that can be done simultaneously or after extracting the juice, or by the rasping of the peel of the fruits before the extraction of the juice. So again, these different processes may lead to different organoleptic properties and chemical composition, of course. So to assess this chemical composition, we used different analytical techniques. You can see that thanks to the team we succeeded to gather in Geneva, we were able to measure the 64 samples of the data set with the GCFID and mass spec. We had 2 runs with the LCMS in different ionization modes, and we also measured the non-volatile part of these oils with NMR. So of course, the idea here is to really get a more complete picture of the physicochemical diversity of these samples by using analytical techniques that will address different kind of compounds. So the first thing we tried to do was to apply a high-level data integration strategy to try to see which one of the data matrices could be used for the discrimination of the geographical origins and the extraction processes. And here, the interesting thing is that when you use different models to build a prediction process, you can collect the prediction from the different models, and using a voting strategy, you can use them to get more robust results using this kind of ensemble learning strategy. So that's what we do. And we were quite surprised to see that actually the geographical origin was quite easily discriminated using single data blocks, single techniques. You can see that with all the different strategies, either GC, LC, positive or negative modes, NMR, all the techniques allowed quite easily to distinguish the geographical origin. So of course, then you can extract biomarkers to see which ones are more related to the different countries, but we had more difficulties to discriminate the extraction processes, and it looks like a single data source wasn't able to offer a sufficient classification of these extraction processes. So the next step, as you can imagine, was to try to integrate this data at the data level with this collection of data matrices and to take advantage of the relationships of the different data matrices that were linked through their observation mode. And with this kind of data structure, you can use different modeling strategy, either unsupervised or supervised, depending on the aim of your approach. And in our case, of course, the idea was to discriminate between the extraction processes, so we used the supervised approach. Maybe first, the first step we can do to try to link mass spectrometry data with NMR is to work with the correlations. And this is something that has been developed quite a long time ago now, and it was called statistical heterospectroscopy with the acronym SHI. And under this exotic name, actually this method is a simple correlation analysis of all the value you can get with a mass spec data sets and the chemical shifts you get with NMR. And you generate this kind of map, which can help to highlight which are the peaks detected in mass spectrometry that are related to different peaks in NMR. And this helped us to get a first information about some of the compounds we found as interesting. For example, here we found that one compound with a given retention time and M2Z ratio value could be related to a series of chemical shifts we measured with NMR. And this helps my colleagues who are experts in chemistry to actually make the difference between these two isomers, because based only on the mass spec data, they were not able to distinguish these two compounds. So they use this correlation analysis to link the peak we measured in mass spec with different chemical shifts, and this allowed one of the two candidates to be excluded with the comparison of the reference spectra of the two compounds. So this can be used to link mass spec and NMR data with simple correlation analysis. But of course, this kind of approach has some limitations. This is limited first to pairwise comparisons. You can only deal with two matrices together. And then correlation, of course, is interesting because you get rid of effects of intensity, but you completely lose this information. And it also may be useful to consider that intensities can play a role for the comparison of these two kind of data, this kind of signals. And this is a univariate parameter, so you will only look at single signals that can be linked together, but I think you quite quickly lose the overall picture of all your signal in the data matrices. So we decided to go one step further with the use of a multi-block analysis strategy that tried to connect the different data matrices. So how does it work? You are very probably used to extract components from single data matrices, and this is done here for each of the data blocks. But then the multivariate model, the multi-block model, sorry, will try to find some kind of consensus space that will rely on a given mathematical criterion to find a common direction in the space of the different matrices to offer an overall picture of all the trends in the data. And of course, if you have a response metric to predict, if you want, for example, to discriminate your groups, you can also add this information to drive the way these components are built. If you think about multi-block modeling, you can think of it as an ecological motto, because you will have to build you have to think global, because you will look for a compromise that accounts for all the data available. But locally, you will use a given mathematical criterion that will extract some kind of variation in the data and that will try to, again, to relate this information across the blocks. And this criterion can be related to correlation, co-inertia, partial least squares, and this leads to the diversity of methods you can find if you want to integrate data sets using this kind of strategies. And then the question is, how complementary or redundant are your data matrices? Complementarity may lead to more overall picture with the different pieces of information that may help to bring a more complete understanding of the system. And redundancy is not always a bad thing. If we think about metabolite annotation, getting different pieces of information about a compound may help to get more confidence in the annotation we can make of it. So again, these two aspects have to be considered, but advantages can be found in the two sides. In the end, you get a multivariate model that will look like a lot the ones you are used to work with a score plot that will give you the distribution of the samples in a new subspace that is common to all the data matrices. The model are most of the time linear, so you will be able to go back to the loadings of the variables to assess their importance in the model. And of course, the additional information here is that you will have access to the importance of the block to compute the different components based on their weights, and you will be able to assess which one was the best or the most informative to get a specific distribution of your observations. In that case, for the analysis of these levanoids, we used an extension of the OPLS algorithm that keeps the ease of interpretation of OPLS with the components that are predictive or orthogonal to the response. And here we have the additional advantage to be able to integrate data from a series of data matrices that are transformed into association matrices that have the same size, and then we end up with a model that integrates all the information on predictive and orthogonal components with scores, loadings, and weights. So what did we get with this modeling approach with our different extraction processes? First, we were quite happy to see that we were able to build a model that was quite predictive with 4 predictive components and 2 orthogonal. And looking at the first principal plane, gathering the 2 first predictive components, we were able, as you can see here on the top left of the slide, to distinguish different kinds of extraction processes with on one hand processes that were related to the rasping of the peel before extracting the juice to get the oil, and on the other end extraction processes that were related to simultaneous extraction or extraction after juice to get the oil. So it was also related to meaningful production processes related to the extraction. The additional information we get to was the fact that the different data blocks contributed to different extents to the construction of the different components. You can see here that the first dimension was mainly driven, so the horizontal one, TP1, was mainly driven by the GC block. So we can imagine that volatiles are important to define this first direction of discrimination. But we were also happy to see that the second components that distinguish the samples in the vertical direction was mainly linked with data associated with NMR. So it means that in that case, probably non-polar compounds were associated with the measurement that we get with NMR. So a nice complementary information obtained with these 2 techniques. And it is not to say that LCMS was not useful, but because as you can see, there are also signals maybe not as predictive as the two other matrices, but there were signals that were measured in LCMS that were very helpful to get more information in the annotation process. And in the end of the study, our colleagues in Firmenich told us that they were really interested in the spumatrice quality of oil, because it's for them it's the gold standard in terms of organoleptic properties. But of course it's expensive, and it's the process that takes the longer. So they wanted to assess what were the characteristics, chemical characteristics of these samples. And since all the data are projected in the same subspace, it's then more, I would not say easy, but it's manageable to extract signals from the different blocks that are associated with it, that are located in the same position in the model, and that are a characteristic of this spumatrice high quality lemonoids. And in that way, we were able to, by coupling information we get from GC, from LC, and from NMR, to get more insight into the chemical characteristics with lower levels in fatty acids and higher levels in sesquiterpenes and fluorocoumarins to characterize these oil of specific high quality. So that's it. I hope I succeeded to interest you in the use of these multi-block strategies to get more insight on your data by collecting multiple sources of information, and to show you that it's possible to get models that account for different data matrices, and that in the end, the interpretation is not that complicated, because if you're used to apply PCA or PLS to your single block data, it's not that different to use extensions that are able to deal with this collection of data blocks. And this is also very useful for biomarker discovery, but because this model remains linear and you get access to the contribution of your variables. I also wanted to end up with some perspectives, because here I showed you that the combination of data was carried out to get a more complete picture of the system. So as on a collaborative mode, but you can also think about these multi-block models as ways to assess which one of the blocks you collected are the best to answer your question. And maybe you could also be interested to use these modeling approaches to in a competition mode to compare the data blocks and select the one or the subset that make the more sense to describe the phenomenon you are investigating. And I just wanted to end up with acknowledgements to all the people involved in this project, here at the University of Geneva, with the two groups of Serge Houda and Jean-Luc Wolfender, people who were in Geneva at that time, but who left and who are still involved in the RFMF, and also, of course, people in our colleagues from Firmenich who also were really involved in the interpretation of the data. And I would like to thank you for your attention. I'll be happy to take questions.
Thank you, Julien, for this talk. I think we have time for a few questions. I can see two already in the chat box, so please feel free to write down your questions. The first one is, do you have an idea why NMR and GC are more potent tools for classification?
I will just go back to this contribution plot. I guess that's the reason of the question. It's probably you have to know that the GC dataset was really carefully manually curated, and the annotation was done with great care. And this was not the case with the LCMS data matrices. I didn't talk about that, but the number of variables was much higher, but that's because there were untargeted and unannotated feature matrices. And this maybe would lead to more noise in the data matrices and the a classification ability that was a bit lost in the middle of signals that may have been filtered better. But I think that this contribution with the polar compounds on the one hand and non-polar ones with NMR is quite interesting to show that we can really gain information by combining different analytical techniques to characterize the samples. But for the chemical composition, I'm not expert enough to discuss this in detail, if it's the question here.
I think I'm gonna ask this question before because I think it's related. And when you have MS and NMR datasets, how do you decide between applying correlation Chi or multi-block integration?
Well, I will rather go for multi-block analysis because you will probably get the same information and more. So if you limit yourself to correlation analysis, well, it's not a bad thing. That's already a good starting point. But the problem is that it's very difficult to associate the links you can highlight between the two data blocks with subgroups of samples or differences between conditions you would like to compare, for example. And this would be possible with the multi-block modeling.
We have also a question about the data integration and the impact of the size of the different blocks. For example, in the transcriptomic data matrices are far bigger than metabolomics.
This is actually a very good question. I also didn't talk about it, but even in our case, the different matrices were of quite different sizes. And actually, there is a normalization step that will scale. You have actually two steps of scaling. You have the standard scaling within each of the blocks. You can apply unit variance scaling, Pareto scaling, the scaling you like that is adapted to your data. And then there is a second step of normalization that will make the different blocks have the same weight, at least at the beginning of the modeling process. They have the same chance to contribute to the model in whatever they size. So there is a normalization step here that is based on inertia or the number of variables depending on the scaling you use. There are different weighing scheme to do that, but it's included.
And two last questions about the one asking about the missing values of TP1 and TP2 in the OPL-SDA multi-blocks. And the other one, wait.
I'm sorry, I'm not sure to get the point because you're talking about missing values on the components.
Well, if the person who asked this question could give more details about this.
Yes,
okay.
I can ask the question better. When you show the OPL-SDA for the multi-block analysis in the slide, well, you didn't put any numeric value for TP1 and TP2?
You mean as in terms of explained variance or things like that? You can get this information, sure. Yeah, you can know which percentage of which was used to build the components. Yeah, that's true. I didn't mention it on the slide, but this is again, everything's linear, so you can get access to this information and know to which extent each of the data block was used to predict which part of the response matrix, as you would get the same results in PLS.
Okay, thank you. Thank you very much.
And last question about the software used for the construction of OPL-SDA multi-block.
This one was implemented in MATLAB, but I know that there are other packages that maybe not OPLS, but there is an increasing number of methods and algorithms that are developed in R if you want to use this kind of models to analyze your data. But this one was developed in MATLAB.
Okay, thank you, Julien, for this presentation and for the answers. I see no more questions, so I think we can move to the next presentation. So our next speaker is Pierre Millard. He is a researcher in metabolomic system biology, affiliated currently to both Toulouse Biotechnology Institute and the MetaTool, part of MetaboHub, the French national infrastructure for metabolomics and fluxomics. His research aims at a better understanding for the organization, functioning, and regulation of natural and synthetic metabolic systems. He develops system biology approaches that combine experimental and computational tools to probe the metabolism functioning with particular interest to exploitation of stable isotopes. Pierre also works on building and validating models of metabolism to investigate the control and regulation of metabolomic fluxes. He then uses this knowledge to understand the role of metabolism in the adaptation of Escherichia coli to its environment, and to design robust and efficient strains as cell factories for innovative and original biotechnological applications. So today, Pierre will present IsoSolve, an integrative framework to improve isotopic coverage and consolidate isotopic measurements by MS and NMR. So Pierre, the floor is yours.
Hello, can you hear me?
Yes.
Yeah, perfect. So thanks for the invitation. So as Julien, I will talk about the integration of MS and NMR data. This time not only about metabolomics data, but more about isotopic measurements. So isotopic information is crucial to understand the functioning of metabolic networks. For instance, if we take as example this very simple network where we have a substrate which is converted into a product of two different pathways for this conversion. In the first one, we have the decarboxylation of the first carbon atom. In the second one, we have the decarboxylation of the last carbon atom. And if we use a substrate which is labeled, for instance, in position 1, the blue dots here, if it is processed for the first pathway, we will have the decarboxylation of the C13 block, and we will end up with a product which is unlabeled. If the substrate undergoes the second pathway, we have a conservation of the isotopic content, and we will end up with a product which is labeled. So by quantifying the labeling in the product or in intermediates, we can identify the topology of active metabolic networks. That is, if we detect labeling, we will know that this pathway is active. If we do not detect labeling, that means that this pathway will be active. And we can access not only the topology of metabolic networks, but also their functioning, that is the fluxes, and we can get some information about the in vivo fluxes. For instance, if we measured 20% of product which contains labeling, that means that this pathway contributes to 20% of the biosynthesis of our product. So this is a widely applied techniques which are useful to study microorganisms, plant cells, tissues, animal cells, and more there are many developments toward fluxomics in human at the whole organ level. So this is important for in systems biology to understand the fundamental principle of life in biotechnology, in synthetic biology, in health also. And the key point in these approaches is to be able to quantify accurately the different isotopic forms of the metabolites. So I will take in most of these presentations the example of amino acids and mainly alanine. So alanine is a C3 compound. For a compound which has n carbon atoms, we have 2 power n isotopic species. So here we have 3 carbon atoms. So we have 8 isotopic species which exist. And what we need is high coverage of the isotopic space. That is a complete set of isotopic species. We need to be able to quantify ideally all of them accurately and precisely. For that, there are different techniques. LCMS, GCMS, and NMR, of course, NMR, proton NMR, C13 NMR, and all these techniques provide different information. So for instance, by MS, we can access the isotopologs of molecules, that is the proportion of molecules which contain 0, 1, 2, or 3 C13 atoms. By NMR, we can access different type of information. For instance, if we look using Toxi sequence, the position of the alpha proton of alanine, we can differentiate the pool of species which are C12 at this specific position and the pool of species which have incorporated the C13 atom. So all the isotopic information we can get is usually partial. For instance, we cannot by MS differentiate this form from this form since they have exactly the same mass. They are partial and they are heterogeneous. Here you can see that what we can measure is always expressed relatively to the total pool of molecules. But for instance, if we apply some HSQC experiments, we can quantify these 4 isotopic forms. But we do not access this form at all. So what we can measure here is expressed relatively to these different forms. And there's been some work in 15 years ago which showed that we can combine MS and NMR information, or NMR information coming from ZQF Toxi and HSQC. So for instance, by ZQF Toxi, we can quantify the proportion of molecules which have incorporated the C13 atom in the second position, and by HSQC, within this pool, we can access the proportion of molecules which are labeled here, but are not labeled in this position, labeled in this position, and so on. So basically, by quantifying, by obtaining quantitative information on each of these set of isotopic species, we can apply some calculations which provide information on the absolute abundance of each of these 4 isotopic species. But currently, there is a lack of informative tools to integrate this partial heterogeneous information. And one of the most basic questions that emerge is what is the current coverage of the isotopic space? That is, what species can we really measure using the different set of methods, LCMS, GCMS, and NMR that have been developed. The second question is, can we improve this knowledge? And for that, what should we measure? And finally, can we, more on a quantitative aspect, can we evaluate the consistency between different types of measurements, for instance, between MS and NMR data? And can we translate these measurements from one representation to another? For that, we need a generic tool and a generic framework able to deal with any type of heterogeneous partial isotopic measurements. And this is what I will present in this. As a test case, we applied the formalism I will present on amino acids because they are ubiquitous, abundant, and central to address many questions in biology. So still with the example of alanine, here you have the isotopic space of alanine, so 8 isotopic species. And the basic principle of our approach is connecting the data to the structure using symbolic calculus. So I will briefly present the approach before illustrating this approach with different examples. So the first point is to map the measurements we have on the isotopic species of the molecule. So, for instance, alanine can be analyzed using 9 different methods, either NMR, GCMS, or LCMS. And for instance, as I showed you before, by playing some HSQC experiments, we can quantify these 4 different isotopic forms. And we can perform such a mapping for all the approaches we currently have. So, for instance, with HSCO deep sea NMR experiment, we can quantify these 4 forms. With GCMS, we can quantify also the form we are having incorporated 0, 1, 2, and so on isotopic species. And each method quantifies specific sets or subsets of isotopic species, and measurements are always relative to the pool of isotopic species that are detected. And this is that we will exploit to integrate the different data. So, for instance, if we want to integrate HSQC and TOXI experiments, so we can just set very simple relationships. So, for instance, this measurement A represents the isotopic species which is labeled only in position 2, so 0, 1, 0, relative to the complete setup of species. And we can do that for all the measurements that are obtained by HSQC sequences. We can also do that with TOXI. So with TOXI, we access this form, so the sum of these 4 isotopic forms relative to the total pool. This is what is expressed here. And we can do the same for this measurement, which is these 4 forms relative to, here again, the complete pool of isotopic species. There is one extra relationship that is always present, is that the sum of all the species of the molecule always sum to 1. And out of that, we have a set of equations that we can solve. And when we solve this equation system, that is when we express the isotopic species as function of the measurement, we end up with these 5 equations, which means that we have a gain of information. When applying only HSQC or TOXI, for instance, we cannot access the absolute pool of this isotopic form, but when applying this formula, this is one I show you in the introduction, we can access the absolute pool of the species labeled in position 2. So when combining these 2 sets of measurements via symbolic calculus, we have a gain of information. Here we can quantify 4 additional isotopomers. Still, the system is not completely determined. As you can see from the first equation here, we cannot quantify each of these 4 species, but only the total pool of the species. And here we do not have any redundant measurements. Now, when we want to integrate LCMS data, we can just add this extra information, map the measurements on the isotopic space, draw the equations which correspond to these additional measurements, solve this equation system, and this time you can see that the system is still underdetermined, but less underdetermined than just with HSQC and TOXI. We can quantify 6 isotopic species on the 8 existing for alanine, and this time we have redundant measurements. That means for some species, for instance, the species doubly labeled in position 2 and 3, we have 2 different formulas which come from 2 different sets of measurements to quantify this species. So, with symbolic calculus, we can connect isotopic data to models and to different representation of this data from the isotopic space of the compound and the structure, basically the chemical structure of the compound. We have implemented this approach in IsoSolve, which is a tool that is freely available in Python on PyPy. That can be used either as a command tool or as a Python library, and it can generate very friendly end-user reports in HTML. I will show you some examples just after. And with this tool, as I will present, we can integrate any type of isotopic data. coming from any analytical source. We can clarify the actual coverage of the isotopic space. We can express the isotopic measurements as isotopomers, chemomers, and EMUs. For instance, I will not go into the detail, but basically this allows transformation of representation between what we can measure in MS, for instance, and what we can measure in NMR. We can also identify redundant measurements, consolidate isotopic data sets, and finally perform some quality control. The input file is quite simple. It's just an Excel file or a tabulated file where you just enter the measurements that you can obtain throughout each of the analytical method, exactly as the table I showed you before. And once we run IsoSolve, we end up with this kind of report where briefly we have the input data, the equations which are derived out of that, the solution, and some status, some information if the system is over, just, or underdetermined, if we have redundant measurements, and so on. And if we have redundant measurements, we can obtain information on the origin of this redundancy. So basically, do we need to apply MS, NMR, and so on to get this extra information? So just a very simple test case. When we integrate all the data we can measure for alanine, we end up with this equation system, and we have the formulas which are drawn where we see that we can quantify all the isotopic species of alanine. Here, the software informs that the system is overdetermined and provide information on the redundant measurements, which can be used, as I will show you, to consolidate the isotopic data sets. So, for instance, regarding the isotopic redundant information, you can see here that two of the measurements can be expressed as a different number of other measurements, which provide information, for instance, on the fact that HSQC is redundant. So NMR is redundant with some information that can be obtained by GRES NMR and GCMS. So if you have these two techniques or these two data sets, no need to acquire this one. As an example, we applied this tool to evaluate all possible combination of data sets we can obtain with alanine, and we based this evaluation on different set of metrics. Basically, the number of isotopic species that can be quantified individually, the number of redundant measurements, and very important information, the gain of information we have when we combine different measurements. So here is a summary of what came out of the study. So here each line is a different combination of each of these measurements. And here we have a summary of the extra information we obtain. So for instance, in the first line in green, we just used data coming from HSQC on the carbon alpha, which HSQC spectra. The second line of just HSQC beta and so on, and we ended up with the last line with all the combination of all data sets. And what we can see is that when we have, of course, single measurements, we have no gain of information. Everything is red, so no gain of information here. But when we combine all the data sets together, we have 10, up to 10 number of redundant measurements and up to 6 isotopic species that can be measured only when the data sets are combined. So out of this analysis, we saw that the combination, the gain of information is dependent on the combination we used. Interestingly, we observed that we have a full isotopic coverage for 40% of the combination with different degree of redundancy. None of the data set is required to cover the full isotopic space, but NMR only proved necessary to reach this goal, because we have in all the data sets, in all the combinations, where we are able to quantify all the isotopic spaces. So to have the full isotopic coverage, we need NMR, but not MS for this example. Another question we addressed is what is a minimal number of methods or data set we need to acquire to have this full coverage of the isotopic space of alanine. And we found the information in the combination number 41, which combined two methods, ATSQC and LCMS, with three data sets. The analysis of the M plus H plus ion by LCMS and of the signal originating from the carbon alpha and beta of the HSQC spectrum. And this gives us this kind of equation system for which we can obtain quantify all the isotopic species. As a test case, we wanted to apply this approach on numerical value, not just on quantitative measurements. So for that, we generated, I will not go into the detail, but we generated the standard sample for isotopic analysis. So basically, we grow a microorganism with a defined carbon source, which has been designed to be able to predict the label incorporation independently of the fluxes. And we measure the labeling in the metabolites, and we can compare the predicted measurement to the experimental values to make sure that we have high quality data. We generated this sample by growing E. coli on acetate. All the information on the production of this sample can be found in this publication. And we sent this sample to different platforms. So the GenoTool and MetaTool platform of MetaWeb, also to the NMR team of the Toulouse Biotechnology Institute, and some colleagues from the Institute of System Biotechnology in Saarland University. And we ended up with this data set. So for alanine, we were able to obtain a total of 21 measurements with different NMR methods, with LCMS and with GCMS. When we want to integrate the data, we have two different situations. First, we have no redundancy. That means that out of the equation, we can directly calculate the abundance of each isotopic species, from the measurements. And when we have some redundancy, we can perform some least square regression to be able to find the abundance of each isotopic species, which explain best the measurement. So this increase the accuracy of the measurements and allow to evaluate the consistency also between the different measurements based on different statistics. I will not go into the detail. So here, this is what we carried out for all the combination data sets, and we perform this analysis based on two metrics: the accuracy. That means the mean error between the expected measurements and the real measurements, and the precision, that is the standard deviation of the abundance of each of the species. And this is what we obtain. So the more method we combine, the bottom part, the better precision we have, and the better accuracy we have. For instance, when we just include the minimal data set we need to cover the full isotopic space of alanine, here in blue you can see the theoretical, the expected abundance of each of the isotopomers, and in orange the one which were determined by data integration. And you can see that for most of them it was very close to what we expected. There was some differences for two of them, but they were not precisely resolved. When we add another data set coming from Toxi experiment, we strongly reduce the error and we improve the accuracy of the quantification. And we combine all the data sets together. This is the situation when we have the higher accuracy and the higher precision, with the mean error below 1% and the mean standard deviation, which is just about 1%. So in conclusion, with this approach, you can improve the isotopic coverage of your metabolites of interest. You can evaluate the complementary of different techniques. So, for instance, do you need to perform some specific development in MS, in NMR, and so on to improve the coverage of the isotopic space? And if so, what do you need to measure? This can be very useful also to consolidate isotopic data set to check the consistency between the different measurements, to convert MS data into NMR data and so on, and to facilitate the interpretation of isotopic data. It's important to mention that this approach does not rely on any metabolic model, just on the structure of the molecule. And we have implemented also, I didn't mention that, but we have implemented a standard representation based on isotopic in keys in IsoSolve to enhance interoperability of the tool in different workflows, databases, and so on towards the FAIR principles. So there are some guidelines on how using and implementing isotopic in keys. I will not go into the details. If you want any information about that, the package, you have all the addresses here about the package, the documentation, the code which is released. Everything is released as open source license. Some Jupyter notebook, for example, on how to use the code, and with the example I showed you on this slide, and the publication from which this data came from. I would like to thank all our collaborators on this work. Of course, colleagues from the meta system of the Toulouse Biotechnology Institute, but also Sergei Sokol from the mat cell of TBI, who has developed the software. The NMR team of TBI, who performed some NMR analysis, also the platform GenoTool and MetaTool from MetaWeb, we carried out some LCMS and NMR analysis, and our colleagues from Germany who carried out the GCMS analysis. And you, of course, thanks for your attention.
Thank you very much, Pierre. It was a very interesting talk. There is a couple of questions in the chat for you. The first one is about the limits in terms of the molecule size. For example, is it easily applied to steroids having way more isotopomers?
This is a very good question. I've not shown this result here, but we applied this analysis on all amino acids. On molecules up to 12 carbon atoms. I think it's close to the upper limit on what can be done with this approach. It takes a couple of hours to compute for a C12 compound. I think we can go up to C15. But it will be difficult to increase above this threshold. We are currently thinking about some developments to improve the speed of the software and increase further the limit. But for now, I would say 15 carbon atoms.
Okay. Thanks. And there is another question asking if this tool was available on workflow for metabolomics, for example, or in any other isotopic studies tools. And if not, are you planning to include them in those kind of workflows?
It's not yet available on the workflow for metabolomics, but it's in the plans to implement IsoSolve. For now, you can use it on your own machines. Everything is available on PyPy. It's very simple to be used, and there is an extensive documentation which is available also online. But we will do that. I hope by the end of the year it will be available on workflow for metabolomics.
Great, thank you very much. I think we are running a little bit late, so thank you again, Pierre, for your presentation. And we will now start with Maëlys. Maëlys Couton, who is a second year PhD student at Nantes Université in the Sésame team. Maëlys, I'm trying to give you the hand in the same time.
Thank you.
And here you go. And so Maëlys is working on the field of position-specific isotope analysis of arginine specifically, and using isotope ratio mass spectrometry, so IRMS, sorry, and her research focused on developing a reliable method to measure intramolecular isotopic composition of arginine, with the aim of providing a descriptors of a flux pattern and urea cycle deregulation. So, Maëlys, the floor is yours.
Thank you, and thank you for this opportunity today. So I will talk about my PhD subject, which is nitrogen 15 position-specific isotope analysis by mass spectrometry and proton NMR. So let's start with the definition of isotopomics. It's the determination of high-throughput isotope profiles or signatures corresponding to global, intra, or intermolecular measurements in natural or enriched abundance to be used for chemometric purposes. So to sum up, it corresponds to isotope measurements in a maximum of stable isotope like deuterium, carbon-13, and nitrogen-15, and it can be used for a real-time monitoring molecule within metabolism. We can measure isotope composition by NMR or by IRMS, so isotope ratio mass spectrometry. And we can find isotopomics in a lot of different fields like archaeology, environment, or in the frame of health and metabolism study. We can measure isotope with different methods. The first one is the global analysis by using an elemental analyzer coupled to an IRMS ratio, so isotope ratio mass spectrometry. Here you obtain the global composition of a mixture, so all the carbon-13, for example, in a mixture. Then you can do compound-specific isotope analysis by GCC IRMS, so gas chromatography combustion IRMS, and you measure the total isotope composition in one molecule. Both of these techniques are very sensitive, but if you want more details on your isotope composition, you can do NMR to have a position-specific isotope analysis. So here you measure the isotope composition on a specific site, like this carbon or this deuterium. However, you have a low sensitivity and you can study only simple molecules, and mainly for deuterium and carbon-13. Why are we measuring isotope in natural abundance? This is because it can occur isotope fractionation during the reaction. So isotope fractionation is a difference of isotope composition in the substrate and on the product. So for example, if we compare two chemical bonds, the bond between carbon-12 and a deuterium will be more stable than a bond between a carbon-12 and a hydrogen. So the reactivity of a carbon-12 and a deuterium will be lower than the reactivity of a bond between a carbon-12 and a hydrogen. So if the yield of a reaction is lower than 100%, it will provoke isotope fractionation, and the isotope composition of the product will be different from the isotope composition of the substrate. So in the frame of health and metabolism study, cancerous cells are able to modify the metabolic pathway in the body in order to produce molecules required for their growth and their proliferation. So for example, in cancerous cell, a previous study noticed that these cells were enriched in carbon-13 and depleted in nitrogen-15. For example, the urea cycle was impacted by these cancerous cells, and it converts the excess of ammonium in the body into different metabolites and into urea. But if we study the molecule of arginine, which is a metabolite produced during this cycle, isotope analysis showed that arginine was depleted in nitrogen-15 in cancerous cells compared to healthy cells. So now we would like to better understand the isotope fractionation which occurred in in cancerous cells. So we want to perform position-specific isotope analysis by mass spectrometry. So to do it, we need to fragment the molecule of arginine in order to isolate each nitrogen of the molecule. So how can we fragment arginine? We choose two different ways. The first one is enzymatic fragmentation. So for example, if we use the enzyme arginase, we can convert arginine into ornithine and urea. So we are able to separate the alpha and delta nitrogen from the epsilon and epsilon prime nitrogen. The yield of this reaction is 100%, and we are still studying the isotope fractionation which occurred in this reaction. The second way of the fragmentation we chose is thanks to electrochemistry. So we use an nickel oxide electrode to fragment arginine, and for now we obtained 3 different fragments. So these structure are still a hypothesis, and we will use NMR for further work in order to have a better structural identification. Now we have our fragment, we will do the isotope measurement. So we inject the different fragments in a combustion oven, where the organic matter is oxidized into gases like carbon dioxide, NOx, and water. Then the NOx will be reduced into diazote, and the water will be trapped. All the remaining gases will be separated on a GC column, and they will enter into the spectrometer. The mass spectrometer is a classical mass spectrometer with an electronic impact ionization source, and the mass analyzer is a magnetic sector. The only difference is that the Faraday cups, in the case of nitrogen, will collect only 3 masses, which are 28, 29, and 30, corresponding to the different isotopologue. So thanks to this machine, we will be able to measure the isotope composition in per mil of the molecule, and it's always compared to a reference gas. In our case, is a bottle of diazote. So to conclude the part of IRMS, it's a really sensitive method, but it's not precise because we need a reference gas. So we want to develop an innovative and non-destructive method combining IRMS and proton NMR for the nitrogen 15 position-specific isotope analysis. And here is the concept. So we do proton NMR because it's more sensitive than nitrogen NMR. And we work in an aprotic solvent to prevent the exchange of proton. And for example, for the case of urea, here is the spectrum. The main peak corresponds to proton linked to nitrogen 14, and the satellite peak here and here correspond to proton linked to nitrogen 15. So after the acquisition, we want to simulate the spectra. So this is a deconvolution with the NMR feed program to extract the area of the satellites and the main peak. The ratio of these areas correspond to the isotope ratio of the molecule, and then we can calculate the isotope composition of the molecule. After this, we would like to draw a calibration curve correlating the isotope composition calculated by NMR and the one measured by IRMS. We did it for 5 pounds of urea, so this one was our standard, and 4 of them were enriched urea. We draw the calibration curve, and we obtained this. The coefficient correlation is not too bad, so this is a promising work for us. We still need to improve the NMR fit, the deconvolution program, but it's promising. So to conclude, we want to perform nitrogen 15 position-specific isotope analysis thanks to two different methods. The first one by using elemental analyzer coupled to isotope ratio mass spectrometer. This is a really sensitive method, so we can apply this method to biological sample, but it requires a heavy sample preparation because of the fragmentation. So we are working on another technique by proton NMR. The fitting program is still under progress, and the sensitivity is lower. So we will apply this to other applications like authentication, for example. But we don't need any fragmentation, so this method could be in the future used as the standard method. So thank you for your attention, and if you have questions, do not hesitate.
Thank you, Mylis, for this presentation. Please feel free to write your questions. I can see one already. How do you plan to improve the fitting program?
For now, we have a lack of repeatability because the area of satellite peak are very small. So we will increase the tolerance, but it's someone in Canada who help us to improve the fitting program. So, but basically, it would be increase the tolerance and have more precise results after the coma. Yes.
Okay, thank you. I think I had the same question. It was about the precision you have when deconvoluting the satellite peaks, but I think it's the same. You're working on that.
Yeah.
Thank you. Okay, so I don't see any more questions, so maybe we can move to the last presentation for this webinar. I don't know if David joined us.
I don't think David managed to join us.
Okay,
I don't see him anyway.
Okay, thank you, Marlene. So I can maybe take. The next presentation will be given by Caroline Peltier, and she's a research engineer in statistics at the Centre des Sciences du Goût et de l'Alimentation at INRAE, ChemoSense platform. She works on developing methods on sensometry and chemometrics, and she works on data integration, specifically using methods, multibloc methods. And she will be giving a talk today about multibloc analysis of aroma release and sensory analysis simultaneously collected in vivo. So Caroline, the floor is yours.
Thank you. Thank you for the introduction and thank you for the invitation. So I'm happy to talk to you today about this talk, which is about pairing aroma release and sensory analysis. So, okay, let's go. So the context is that where I work in a center of taste behavior, we have several dynamic methods to measure what happens during a tasting. We have some chemical methods, which are, for example, PTRMS, which consists in measuring volatile molecules directly in the subject's nose, keeping the kinetics. And we also have temporal sensory methods, which are called TDS or TECATA. I am going to develop that later, but which consists in asking the subject to score dynamically its sensations. And the questions of the studies that I'm going to present was, are there differences between pairing PTR with TECATA or PTR with TDS, these two sensory methods? So first, I'm going to present you the data set. Then I'm going to present you a multi-block method, which is called RGCCA. And then I finally will apply this method to our data set. So regarding our example data set, it was collected in 2090s. And And so 3 chocolates were tasted by 16 panelists. So we had A, B, and C, which are chocolates. They were previously characterized by a profiling task. So A was shown to be fruity, B was shown to be spicy, and C was shown to be cocoa and woody. And so the 16 panelists have done some tasks. So they were connected to Peltier RFMF device, and then they did either TDS or T-kata task. And so, for example, for one given evaluation, we have this procedure. So we connect the subject to the Peltier RFMF. There is a full blank period of 30 seconds around to check the breathing and things like that. Then the subject can put on start and put the product in his or her mouth. And then he has to do the task TDS or T-kata. So now I am going to explain what it is. So let's imagine that I taste a chocolate. I put the chocolate in my mouth, I click on start. Then I feel that there is some dry fruit aroma, so I click on it. Then I feel that finally it is more spicy, so I click on it. And then I don't feel anything and I click on stop. So I have these sequences when I have selected only one descriptor during each time. So this was TDS and T-kata. In fact, the difference is that it's kind of the same thing, except that you can click on several descriptors at the same time. So for example, I put the chocolate in my mouth, I click on start. I feel some chocolate, some cocoa, I click on it. I say that cocoa is not the only one descriptor in my mouth. There are also woody descriptor. I click on it. Then I can unclick woody if I don't feel it anymore. I can say cocoa again, and then I click on stop. So there are really 2 different methods which are alternatives to declare your sensations. So we obtain some data which are kind of complicated because they are temporal. And so as they are temporal, we have to summarize them. So for TDS on T-kata, we have chosen to keep only the duration per descriptor. And for Peltier RFMF, we have chosen to keep the area under the curve per molecule after having corrected the breathing. So here, for example, I have this kind of data. I have 4 blocks of data. The first one is TDS. I have one evaluation for one subject and one session, with each column correspond to a sensory attribute, a sensory descriptor. Same with T-kata, and for Peltier with TDS, in columns it is intensities of molecules. So here we obtain some multi-block data. And now we have to deal with that and with the fact that maybe we will have a lot of molecules here and not so much sensory attributes in the sensory blocks. So how to do that? I am going to try to explain you what happens to begin with only 2 blocks of measurements. So let's imagine that we have only 2 blocks to deal with. In this case, we have methods that exist that we know, which are, for example, PLS, partial least square regression, and canonical correlation analysis, so CCA. And in fact, the objective of PLS is to maximize kind of a covariance criterion between 2 scatter plots. So this is the criterion of PLS, and you have some constraints on the weights that should be of norm 1. And besides, you have CCA, which aims to maximize the correlation between the 2 scatter plots. And in fact, maximizing the correlation could be equivalent to maximize the covariance with some other constraints here. So in fact, you can see that here the criterion is the same. So as it is the same, the idea would be to merge these 2 methods with the use of a parameter, which is called tau. And you can see here that we can use a maximization criterion of the covariance here with this kind of constraint, which is simply very naturally the linear combination of this constraint and this one. So you have kind of a continuum between PLS and CCA, choosing a two parameter. So this was for 2 blocks. Now we have G blocks with G higher than 2. So we have a lot of possible covariances. So how to do with that? How to combine the different covariance between the different blocks? The very natural idea, again, is to sum all these covariances. But maybe you will have some negative covariances, so you will want it to look maybe to the absolute values or the square values or things like that. To do that, we choose to use a convex function that could be that could do that. This function is called G here, J. And so this is the first thing. And the second thing is that some links could be not relevant between these blocks. So you can choose to not select some covariance term. That is to say that we added the parameter C here. That is 1 if the blocks are connected and 0 if else. And finally, we have this criterion here, which is the natural extension of what I have shown before with the extension here of the constraint. So with this criterion, we can do a lot of things, and it is regularized generalized canonical correlation analysis, which is a paper of Arthur and Michel Tenenhaus. And this paper is very interesting because, in fact, you can look at with this framework, you can get back a lot of existing multi-block methods with a single algorithm. And this algorithm is shown to be convergent. It's a NIPAS algorithm, that is to say that you can use it with some missing values. It's freely available on GitHub, and recently it was updated on the CRAN. So it's very nice to use it. So let's do it on our data. So I have 4 blocks with the same individual, so that's very important. We have an horizontal structure of multi-block data, and we choose to select a criteria, which is to maximize the covariance, the sum of the covariance between PTR TDS and TDS, because they were simultaneous, and PTR T-kata and T-kata because they were also simultaneous. So what does it mean? That means that the connection between the block correspond to this matrix. So you can see that here you have 1 when the blocks are connected. So TDS here is connected to PTR TDS and not to the other blocks. And the objective of this criteria is that we try to explain the sensory durations by the ion intensities. So what we have obtained is that kind of results. So on the left, you have the individual maps that correspond more or less to it's like a PCA map. So here you can see that I have colored the points, the evaluations, according to the product to which they belong. And so here you can see that on the first axis, you have the C product here, which is discriminated from the 2 other ones. And this is true for every blocks of data. So that's the first thing. Then you can see on the second axis, then A and B are well discriminating for all maps, but except for T-kata here, which is not really well discriminated. So here we can observe a difference between T-kata and TDS, and T-kata seems to be maybe more difficult to discriminate A and B than TDS. And on the right, you have the weights obtained for the first axis, so only the horizontal one. I didn't have the place to put the second axis one. And what you can see is that this one corresponds to this one to this one, and so on. So here what you can see is that, for example, on the first axis here, I will have the molecules, this one and this one, that are more present in the product C, which is So here you can see, oh, sorry, you don't see that. Okay, with my mouse here, you can see that here all that molecules are likely to be correlated to C and to woody and cocoa, yeah, and not to fruity. So here we have this conclusion for A and for these two graphs, and so we can see that in fact we have the same conclusion looking at what happened in Tecata. So you can see that here these are the same molecules here, and the same with the same attributes that are shown here. So to conclude, finally, were there differences between pairing PTF Tecata and TDS? There were no big one. It seems that TDS was more likely to discriminate better the products and to be maybe kind of better in our experimentation. Regarding OGCCA, it was shown to be a very powerful tool to deal with multi-block data. In fact, it includes and generalized most of the usual analysis. You can find back PCA for unsupervised analysis, PLS for supervised method, but also, for example, multiple factorial analysis or hierarchical PCA or so on. So you have further options in the CCA. You can do superblock options. You can use sparsity, that is to say that you can do variable selections. And also in the new version of the package, you can also do some cross-validation, bootstrap, and so on. And to conclude, I would say that here we are focused on durations and intensities, but other methods could be considered to take the temporality into account. And it's something that is really interesting and that we would like to look at for the next month. Okay, thank you very much for your attention. And if you have any question, I will be happy to try to answer it.
Thank you very much, Caroline, for your nice presentation. I was sure that Julien was going to have some questions, so thank you for the very interesting talk. So he has several questions. I might ask them one by one. The first one is, did you account for blocks involving the same attributes like TDS and TCA-TA?
I'm not sure. Could you repeat? I'm not sure to understand what it means.
Oh, maybe Julien, maybe if you want to ask your question directly, it might be easier.
Thanks a lot, Caroline. You showed us that you use the design matrix to define the relationships between the blocks, but I had the feeling that it was related to the observations only. Is it possible to do that? I guess it's possible to do that with the variables, because the TDS and the TCA-TA blocks involve the same attributes.
Yeah. What you mean is that, in fact, I could see it as a cube structure. Is that what you mean?
I guess it's kind of complex because it's a partial cube.
Yeah, partial cube. Yeah. It would be nice to try to do thing like that, but I didn't try it. But you're right. In fact, here, as I have the same names, I could see that as a tensor and see that as a tensor, and then related have a very funny structure, in fact, of data, and dig it like that.
Thanks. So you showed us the observation connection matrix, actually, but there is another one with the variables, or how does it work?
For this one, okay. For this study was an example. So here, in fact, I have considered this connection matrix. So I have my 4 blocks. I've considered that there were totally horizontal structure.
Okay, so it's only for the observation. Thanks.
Yeah, only a horizontal structure, as you said previously. So
I'll read my next question if I can. Marine, thanks. Did you investigate the effect of the tau parameter? Because you mentioned that you can get CCA or PLS results or something in the middle. How did you manage to set this tau parameter?
So here I have not I have just put tau equals 1 to be because I wanted to maximize the covariance parameter. I am going to just get back here. So yeah, so here I was more in that case. So I have not optimized the tau parameter, but we can optimize it. In fact, it's implemented in the package. If you want to implement, if you want to find out the best tau to obtain, for example, a correct ridge approach, you can do it. But for my presentation, I used tau equals 1 as in usual PLS.
Okay, thanks. Then I guess that the next challenge is to define what is the best results, since here you're in an unsupervised approach. So.
In fact, this challenge was already kind of answered with a paper of Streamer and Schaffer, I think, something like that, and it's already implemented, but it's more to optimize things in the canonical correlation analysis, because here you can see you have the variance here, and sometimes you have matrices that are not of full ranks. And so this is why this structure here is very interesting, because you can see that it looks like a ridge approach. And so you have something which is not of you have here, this is not a full ranks, and it is a full ranks, and so you can deal with this tau, and so we have an optimal tau that is a parameter that you can enter in RGCCL.
Okay, thanks. And my last question was related to the way you were able to associate subsets of variables to groups of samples with a p-value and guess resampling somewhere. Do you know? Can you explain how it works?
I'm not sure to understand the question. Here, in fact, I have not put the product information in my data. It's not supervised by a product. Was it what you?
Because you have significance values on the right that are related to.
Yeah, you can. Okay. Right? Okay. Sorry. Yeah. In fact, here it is some bootstrap. We did some bootstrap experimentations on the weights. So that is to say, then you run RGCCR one time, and then you do bootstraps, and you obtain. Here I made 1,000 runs, and with these runs I obtain confidence intervals that shows here that they are really different from zero, and in fact it's kind of a t-test here to conclude to the significance of the weights. Was it clear? Or?
Yeah, sure. Yeah,
okay.
I thought it was a way to associate specific variables to subgroups, but maybe I didn't get it right. Thanks a lot. I think that we should definitely have a closer look at this RGCCA package.
Yeah, with pleasure.
Cool.
Great.
Thanks.
Thank you very much, Julien, and thank you for Caroline for your presentation and taking the time to answer Julian's question. So thank you again to all the speakers and to all the audience who joined us today. And I will let Gina conclude with a little announcement for the next webinar.
Yes, thank you, Marine. I just sent the information in the chat box. So our next webinar will be held on the 7th of September at 2 p.m. Paris time. So the theme will be metabolomics applied to health studies. Our plenary speaker will be David Wishart, and it will be the only guest for this year's webinar series from outside of the French-speaking metabolomics and fluxomics network. 3 other presentations will be given by Ariana Sirio from Liège University, Jessica Micheletto from Laboratoire d'études du métabolisme et du médicament in Saclay, and Marine Leterte that you met today from Sésame Laboratory in Nantes. So save the date, it's September 7. And I hope you will all have a nice summer vacation and hope to see you all joining our next webinar. And thank you for joining today's webinar also.
Thank you. Bye, everyone.
Thank you. Bye-bye.